was mainly looking at this chemokine receptor called CXCR3, and it looked at it from both structural and pharmacological perspectives. So what's really interesting about this receptor is that in humans it exists as two different isoforms. And in some disease contexts, those isoforms can have opposing functions and physiological roles. So from my PhD, I already asked the question, how can we understand the differences between those isoforms, and therefore how can we target those differences to design isoform-specific drugs at this receptor? So CXCR3 belongs to a family of G protein coupled receptors. And those receptors share a lot of common features. And of them, you have 7 transmembrane helices. You have 3 extracellular loops, as well as 3 loops on the intracellular side. So when those receptors are activated by an agonist, so that could be either a peptide or a neurotransmitter or even a synthetic agonist, those receptors undergo a conformational change. So this change involves the contraction of helices 5 and 6 together. And when that contraction happens, there is a space created in the receptor for transducers to bind and interact with it. So those transducers can be either G proteins or beta arrestins, and they then trigger different physiological responses down the line, and that response will differ based on the transducer recruited. So as I said, CXCR3 is a GPCR, and the reason we want to look into it is because it's important in immune function, and its dysregulation has actually been linked to several autoimmune conditions and cancers. So how does CXCR3 exert its role in immune function? So when there is an inflammatory stimuli, so that could be, for example, tissue damage, inflammation, tumor growth, or infection, this sends signals to T cells to release something called interferon gamma, which then releases 4 different pro-inflammatory chemokines. And at the same time, your T cell, which normally has a bit of CXCR3 on its surface, starts upregulating it. And those chemokines basically create a chemical gradient in which they attract the T cell expressing CXCR3 to be in close proximity to them. And that usually occurs at the sites of inflammation. So when these 2 components are close together, they can obviously bind, and they undergo the change that I described in the earlier slide, where they can recruit either G proteins or arrestins, and therefore trigger different signaling responses down the line. So for G proteins, that will be T cell chemotaxis, and for beta arrestins, that can also lead to inflammatory response, but more often it's also associated with receptor degradation and signaling. So actually, CXCR3 can be quite useful because in normal physiological response, T cells are directed to through this movement called chemotaxis into the inflammation sites and help resolve the inflammation altogether. But during sustained inflammation, so for example, in conditions such as cancer, T cells get continuously recruited to the sites of inflammation. And when this happens, T cells can start attacking healthy tissues, and that can lead to chronic inflammation and therefore disease progression. So in sustained inflammation, this is where therapeutic antagonists have a potential. So under this sustained inflammation, if the chemokine binds to the receptor, antagonists such as monoclonal antibodies, for example, can block this interaction between chemokine and CXCR3. They can therefore stop this continuous recruitment of T cells and therefore stop attacking of healthy tissues, chronic inflammation, and disease progression. But this is where things get complicated because CXCR3 exists as 2 isoforms. And these isoforms are pretty much identical when we look at their transmembrane sequences, but where the difference happens is it's in their N-terminal region. So CXCR3A is what we would call a shorter isoform, and CXCR3B has this N-terminal region elongated by around 50 amino acids. And this region is important because that region is what first recognizes agonists. So it can influence how the receptor interacts with its endogenous chemokines as well as therapeutic ligands. Besides this difference, those 2 receptors have also different expression profiles. So when we look at CXCR3A, it's mostly expressed on T cells, endothelial cells, fibroblasts, and epithelial cells. But CXCR3B doesn't really have expression on T cells, and it can be mostly found in endothelial cells, fibroblasts, and pericytes. Additionally, another difference between them is that CXCR3A can be found mostly on a cell surface, whereas CXCR3B has a bit of a mixed distribution. So, for example, in pericytes, it can be found on cell surface, but in endothelial cells, it can be found both in the cytosol, but also on the cell surface. So, CXCR3A and B, besides those differences, also trigger different physiological and signaling responses. So CXCR3A is the better characterized of the two isoforms, and is known to signal for G-alpha-IO proteins and beta-arrestins, although we don't really know which subunit of G-alpha-IO it happens for. And then the physiological response that CXCR3A causes can be T-cell chemotaxis, which occurs mostly through G-protein-mediated signaling, and beta-arrestin triggers responses such as tumor proliferation and metastasis. So because of that, CXCR3A has been actually found to be upregulated in patient samples in a lot of autoimmune diseases and cancers. So for autoimmune diseases, found that CXCR3A is most correlated with, for example, RA, multiple sclerosis, and type 1 diabetes. But it's also quite prevalent in a lot of cancers, with colorectal cancer, prostate cancer, and breast cancers being some of the examples when this receptor is overexpressed. So CXCR3B is not so well characterized. We don't really know how it signals, although it's been reported to trigger P38 MAPK pathway down the line, which is associated with cell apoptosis. So that's obviously quite good because that means cancer cells can die. And therefore, it's been correlated with positive prognosis of cancers that CXCR3A is involved in. But majority of therapeutic efforts don't really consider the CXCR3B isoform. And that's because a lot of the therapies that have been in experimental phases or clinical phases are small molecules. And as you can see by these two examples here, both of these small molecules bind within the transmembrane core and transmembrane regions of the receptors, which don't really differ between these two isoforms. So because of that, small molecule therapies potentially risk simultaneous inhibition of both isoforms, especially in tissues where both are expressed at the same time. And one example of these tissues is, for example, breast cancer stem cells, where both of these receptors can be found in different ratios. So preferential inhibition of CXCR3A over CXCR3B would be desirable in that context. So because of that, this project aimed to really understand the pharmacological differences between how those two isoforms interact with their endogenous ligands, but also understand how they can be inhibited by different antagonist classes. And by that, I mean different small molecules and monoclonal antibodies. We also want to understand how they differ structurally, and we want to do that to help guide selective targeting of one isoform over another. And we will also do that by integrating structural and mutagenesis approaches. And the big picture aim here is to inform rational and isoform selective drug design strategies for treatments of autoimmune diseases and cancers. So the first question that we ask is what differences at a molecular level and signaling level might underline opposing functions of these two isoforms. So in order to understand the pharmacology of the receptor, we conduct breath-based assays. So using beta-arrestin as an example, we attach a luminescent flag onto the receptor, and we also attach a fluorescent tag onto the beta-arrestin. So when the agonist binds, the receptor and arrestin are in close proximity together, leading to high breath signal, which could be then plotted based on concentration responses, with lowest concentration here and highest here. And that can then be extrapolated as area under a curve of each of these different concentrations, giving us a concentration response curve, which will help derive parameters such as potency and efficacy. For G protein recruitment assay, the principle is similar. However, here we attach the luminescent tag on the G alpha subunit and fluorescent tag on the beta gamma subunit. So when the agonist binds, the GDP gets phosphorylated to GDP. There is dissociation between these two subunits, and that leads to lower Brett ratio, which similarly can be plotted in the same way as a concentration response curve. So when we look at pharmacological characterization of CXCR3A isoform, we simulated this receptor with 4 of the endogenous chemokines and measured how it responds to G alpha IO proteins. And what we saw from here is that this receptor is not really selective, and it recruits 3 of the chemokines quite well, except CXCL4. And then when we measured this at beta arrestin, we saw that the profile is also quite similar, except CXCL9 is recruited a little bit, recruits beta arrestins to a lesser extent. And the other thing that we saw here is that the order of this recruitment pretty much in all cases of arrestin and G protein pathways followed CXCL11 being the most potent chemokine, and then CXCL10 medium, and L9 being the weakest. So we looked into the literature, and there is actually some structural explanations to help us deduce why ligand pharmacology at CXCR3A is what we observe. And there is 2 major factors that contribute to it. The first is the CXCR3 N-terminus itself can modulate chemokine-mediated signaling and binding. And what we can see just from structures here is that the N-terminus of CXCR3A wraps itself around CXCL11 and forms more stable interactions. However, by Cryo-EM, the N-terminus with these other 2 ligands wasn't really resolved, potentially indicating that there could be some conformational flexibility in that interaction. Another factor is that the chemokine N-terminus itself, so that's the ligand, and its hydrophobicity affect the strength of the interaction with the transmembrane core. So when we first look at the depth of insertion, we can see that CXCL11 is actually inserted the deepest among all of these other chemokines. And then when we zoom in onto the binding pocket, what we can see is that this binding pocket is quite hydrophobic. So therefore, something that's also quite hydrophobic and has a lot of aromatic or phenyl rings will be more stabilized within it rather than something that's just polar. So as we go from CXCL11, 10 to 9, we can see that the hydrophobicity of this N-terminus actually decreases, and that means that the strength of the interactions also decreases as we go from left to right. Then if we want to see how this N-terminal extension that's present at CXCR3B impacts the strength of this signaling, when we measure the G protein recruitment, what we saw is that all of the G alpha IO proteins are recruited, but only in response to CXCL11, and the other ligands only recruit these transducer upon highest concentration. And the same was seen for beta arrestin, where CXCL9, 10, or 4 actually has not recruited arrestins at all. So from then, we can see that CXCR3A is more promiscuous receptor, whereas CXCR3B is more selective, being more selective against CXCL11. So what structural differences underline this unique pharmacological profile for CXCR3B? So to do that, we wanted to characterize CXCR3B on a structural level. And to date, the only existing CXCR3 structures have been resolved in active bound and inactive states, and those inactive states are with small molecule antagonists. But no structure of CXCR3B exists. So since CXCR3B responds selectively to 11, we decided that this is the complex subcapture. And when we looked at our data, we saw that G alpha I1 was the transducer recruited quite potently, so decided to use that for structural studies. So we hope that solving this DNGI1 CXCR3B CXCR11 structure would allow us to see what's different about CXCR3B at the molecular level and hopefully connect those differences to distinct signaling hopefully also map why is that happening and what is the correlation to the opposing physiology, with the ultimate goal to guide drugs that target only one isoform and not both. So this is the workflow for biochemistry and Cryo-EM. So this was the construct we used. We had CXCR3B. We didn't really introduce any mutations. However, there was some purification tags and a tag that helped optimization, expression optimization in the insect cell system. We then introduced these contracts into baculoviral vector, made virus, transfected insect cells to have pellets that express a lot of protein. For G protein, we have G alpha I1 with dominant negative mutation to sort of help stabilize the G protein heterotrimer and prevent its dissociation. And we also have a fused beta gamma construct. And in the same way, we make virus from that, put it into the insect cells. So they express a lot of this protein. We then solubilize these two components together. And we do so in detergent micelles in the presence of the ligand. And then go through the standard purification where we pull the protein by flag and separate it by size exclusion from all of the aggregates. So this is the size exclusion trace. Obviously, we have quite a huge void, but we also have a peak that's corresponded to the complex by Western blot and plumacy stain. Put it on negative stain, and we thought this looks quite good. For reference, this is what you want to see on the negative stain images. So a little micelle here and two little dots protruding from the bottom of it, which are the G proteins. So thought that this is good to put it on the grid. So put it on the grid for Cryo-EM, collected around 4,000 micrographs, classified that, motion corrected, and all of those processing pipelines that put it into those 2D image projections. From that, we were able to build an ab initio model, which obviously doesn't look great, but then we've done a few steps of refinement and polishing that finally gave us something that had a bit of shape where atoms could be modeled into. So this is the structure that we got, and from what we see is we got the receptor in the micelle with 7 transmembrane helices, which is typical of GPCRs, and we also saw the G alpha protein inserted by the C-terminal tail into the receptor's intracellular core. And we also saw densities for the G beta and G gamma. From that, I was able to model the residues and come up with the structure. And what we saw is we could model the extracellular loops, which are quite flexible. I saw this canonical beta-RP shape in extracellular loop 2. And also, when I looked closely at the G protein interface, I saw 6 hydrogen bonds that stabilize the G protein and the receptor together. However, the main limitation that I found is, despite seeing that this receptor is obviously in active state and is G protein bound, I wasn't able to really capture the density for the N-terminus, which is what differs between these two isoforms, and therefore I also could not really see any density that's responsible for the ligand. And this was quite interesting because obviously a lot of ligand was present throughout all the purification and also sample preparation. And we also from pharmacology know that GI1 recruitment to CXCR3B is ligand dependent, and the literature reports that this receptor is not really constitutively active. So what could account for what we observe in this lack of ligand density in the structure? So one explanation could be that CXCL11 binding kinetics can be unfavorable and lead to fast dissociation rates of CXCL11 from the receptor. So even though if throughout sample preparation we keep on priming the receptor with the ligand and the complex could transiently form, CXCL11 could dissociate from the receptor over time, and therefore this could be what we see. And the reason that the complex stays intact even without the ligand could be because we have those mutations within G protein that prevent the dissociation. However, another possibility is that CXCR11 could make interactions with a flexible epitope instead, which is something that we couldn't resolve in cryo. So when we look at CXCR3A, we could see that the N-terminus is quite ordered, that it forms a stable interaction with the ligand, and there is overall less conformational heterogeneity at the extracellular phase. But for CXCR3B, the N-terminus is obviously significantly disordered because it's elongated and it's outside of the transmembrane, inside of the membrane. It is the ligand epitope might not be as stable, and this N-terminus could also add extra conformational flexibility. And this is also seen because when we did our local resolution estimation, the regions outside of a little bit protruding outside of the transmembrane core that are on the extracellular side had lower resolutions, potentially also indicating that there is some flexibility in there that's not captured by cryo. So could CXCR3B structures still tell us about isoform-specific differences despite the lack of the resolution? So when we look at the pharmacology data again, we can see that both of these receptors are able to recruit G-alpha I1. However, CXCR3B does so CXCR11 recruitment of G-alpha I1 occurs with 5-fold lower potency at CXCR3B. So by comparing the two structures of CXCR3B and N, we instead want to now look at how would this extended N-terminal difference propagate to the geometry of the receptor core, and how the differences at the molecular level and conformational level within this core can then propagate to the G protein interface that could account for this decrease. So we overlaid CXCR3A and B together, and they were quite aligned, and the RMSD actually shows there is minimal deviations between them. However, some prominent differences were observed. So the first difference I would like to point out to is the transmembrane helix 1 points a little bit outward at CXCR3B compared to A. And this is interesting because, as we know from the transmembrane helix 1, this is where the N-terminus protrudes out of. So it is possible that this N-terminus could add some conformational strain that makes the helix itself more flexible and makes it swing outward. Another difference observed was that TM6 and TM5 were more contracted together at CXCR3B, and this could be important because those helices and the opening between those helices is important in receptor activation, as I showed before. We also saw that the extracellular loop free at B is pointed slightly more inward, and this is important because for a lot of chemokine receptors, the extracellular loop forms a part of chemokine recognition phase. So the fact that it's in a different conformation could potentially be telling a little bit about dynamicity or how stably the ligand is bound. Another difference was the intracellular loop 1 was pointing further away from the G protein heterotrimer. And this is important because it's obviously further away. So there is less interactions, stabilizing interactions it can form with the G proteins itself. So the next thing that we compared was more at the atomic level. So before I show all of the residues that we looked at, I want to just quickly explain that chemokine binding is quite conserved among this receptor family, and it usually occurs with a two-step binding model. So in this model, chemokine is first recognized by the N-terminus here. So that is referred to as the initial docking and the recognition of the chemokine. And then this N-terminus wraps itself around the chemokine core, inserting it into the transmembrane helices and upper some extracellular loops, thereby stabilizing it in there. And this is called triggering. And the reason it's called that is because this then triggers certain molecular microswitches that make the receptor adopt this conformational change, leading to the opening of TM5 and TM6, and therefore allowing for G proteins to combine. So when we just compare CXCR3B, the structure we got in pink here, to an inactive state CXCR3A structure, we could see obviously a clear difference in the rearrangement of transmembrane helix 6. And then when we zoom in on the conserved activation microswitches, which are pretty much similar for all class A GPCRs, we see those movements that are conserved with that. So for example, on TM3, that's the dry motif arginine that is pointing a bit upward. The tryptophan in TM6 is pointing down from the extracellular side, and the tyrosine on TM7 moves away from TM6 to allow for the opening to happen. So now when we look at this on a molecular level, when we look at chemokine recognition site 3, we can see that pretty much all of these residues align quite well together between the two receptors isoforms. But at CRS2, there is some differences, and the difference that I really want to point out to is this tryptophan 6.48 here, and that's because this tryptophan is a part of these microswitch conserved motifs that ultimately dictate receptor activation. And this position was also supported by the Cryo-EM map. So this is CXCR3A map, and we clearly could see sort of an orientation that's parallel to the backbone amide chain. But with CXCR3B, we see that this tryptophan lays a little bit flat. And that was also the only difference in activation microswitch geometry that we saw among all of these conserved microswitch motifs. So it could potentially be indicating of whether the ligand, how dynamic the ligand is, or if it's stably bound at all. So then when we extend these differences to looking at the G protein interface, what we see here is that CXCR3A, the alpha 5 C-terminal helix, is inserted slightly deeper than in B. And then when we overlay them together, the alpha N helix of the G protein is pointed slightly outward from the receptor in CXCR3B. And then when we zoom in at the interactions, we can actually see that CXCR3A forms 8 hydrogen bonds and 160 van der Waals contacts, that CXCR3B forms 6 hydrogen bonds and 74 van der Waals contacts. So that's obviously a smaller number, although a few of these interactions are still conserved. But overall, this decrease in stabilizing interactions could potentially be related to what we observed in pharmacology, where the potency that CXCR11 has at GL5 recruitment is a bit lower for CXCR3B. So just to summarize those structural and pharmacological observations, we saw that CXCR3A is a promiscuous receptor and it recruits CXCL11, 10, and 9, with CXCL11 recruited robustly. All ligands allowed the receptor to recruit both G proteins and arrestins. CXCR3A observed a stably bound ligand at the transmembrane core. We saw that TM1 was closer to the helical core as well. ECL3 swings outward. TM5 and TM6 were further apart, and overall it had more stabilizing interactions at the G protein interface. When we extend that to CXCR3B, we could see that it selectively binds to CXCL11, which recruited both G proteins and arrestins. We could see that the extended N-terminus wasn't really captured, and it's possible that it could occlude or create a potentially conformationally larger flexible site for the ligand to bind. We also saw several conformational differences, such as extracellular loops swinging more inward, TM1 being more disordered and further away from the TMV, and TM5 and 6 being more compact. And obviously, overall, less stabilizing contacts at the G protein interface at CXCR3A. So now that we looked at it, and obviously, as I said before, that CXCR3A is the receptor associated with the disease progression, this is obviously the isoform that we want to inhibit enzyme. So we asked ourselves the question, how can CXCR3A be selectively targeted over CXCR3B in drug design? So currently there is no clinical approved drug for CXCR3 on the market, and that's despite this receptor being involved in a range of autoimmune and cancerous conditions. However, there's been some successful contenders. So one small molecule AMG 487 actually reached the furthest in clinical trials, reaching phase 2 for MS, psoriasis, and RA, but was terminated due to metabolite instability. However, as I mentioned earlier, small molecules also can't really distinguish between the two receptor isoforms, and that could also contribute to their ultimate lack of success in clinical trials. So this is where monoclonal antibodies come in, because they can allow for higher selectivity, not only between receptor isoforms, but also between structurally similar GPCRs. So one antibody is MAP 160. It works by depleting CD4 and CD8 positive T cells. It basically has been found to inhibit ascites-directed tumor cell migration and was used and intended for treatments of myeloma and ovarian cancers. Another monoclonal antibody works pretty much in a similar way, in the way that it depletes T cells. However, it's more designed for the treatments of autoimmune diseases such as psoriasis and vitiligo. So we wanted to measure how these antagonists interact with their receptor isoforms. And to do that, we used operational model of allosterism. In this model, we basically measure the measurement called KD, which is a functional affinity of the allosteric ligand. So it measures how binding of the antagonist affects the agonist binding. And then we also have a parameter called log alpha beta, which measures cooperativity. So that basically means how the effects, the allosteric effects, the antibody exerts on orthostellate ligand binding, propagate down to how it inhibits the agonist-mediated signaling. And in general, more negative log alpha beta means more negative cooperativity, so more negative allosteric modulation. So when we look at the small molecule AMG 487 and we measured inhibition to CXCR3, since that's the shared ligand of both isoforms, we saw that there was no major differences in log alpha beta, and the small molecule inhibited both of the isoforms quite well. And this is because it's not surprising, given that the small molecule binds within the conserved transmembrane core, which doesn't really differ between CXCR3A and B. When we then extend that to monoclonal antibodies, we see that overall the degree of inhibition was higher at CXCR3A versus CXCR3B, and we saw more prominent differences in log alpha beta. And the same was seen for MAP 160, which, for example, wasn't able to inhibit G-alpha I1 recruitment at all at CXCR3B. And the reason for that is because MAPs obviously are significantly larger, so they won't be able to interact within the same transmembrane pocket as small molecules. And they interact with those larger and more diverse epitopes that are on the extracellular side, including the receptor N-terminus. So since overall antibodies inhibited CXCR3A signaling more selectively than B, this could potentially give potential for achieving isoform selective modulation. So can we determine the binding epitope of a monoclonal antibody at CXCR3A isoform to aid selective drug design? However, the Cryo-EM attempts at elucidating this epitope were not successful, because even if the complex was purified well and separated from aggregates, and we could see it in negative stain 2D classes, so you can see a receptor top view and a little density protruding out of it responsible for the antibody density. When we put this on Cryo-EM, there was not really much particles of interest, and when we did 2D classification, we found just sort of empty receptor micelle, and we found no evidence of antibody binding. So because of that, we tried to pursue different approaches to determine this epitope. And we did that in collaboration with our partners at Servier, who helped us with molecular dynamic simulations of the AlphaFold predicted model of the Fab fragment of this antibody put within the receptor. From then, we tried to analyze specific interactions. So by that, I mean interactions that are not on the main chain of the receptor, or on the side chains, and also interactions that occur in more than 10% of the frames. So we acquired 1,000 frames total. This was done over 30 nanoseconds, and every 30 picoseconds we were acquire a frame. So we found one residue at the N-terminus that has formed quite a lot of interactions with FAB heavy chain. We also found a residue at transmembrane helix 1 and ECL1 contributing to it. We also found 2 residues at extracellular loop 2 that formed quite a lot of interactions in a large portion of the frames. So then how did we, once we had those residues, the next step was to computationally validate them, and we chose TruePAP system for validation. So how it was done is each residue that was appearing in more than 10% of the frames was individually mutated to alanine. So we chose alanine because that's chemically inert. It doesn't really have a side chain to form interactions, and then substituted each residue with that. The pathway that we wanted to inhibit was the inhibition of CXCR3A following the stimulation with CXCL10 ligand at G-alpha I3 pathway. And the reason we did that is because that had the larger signaling window. So we thought that would obviously give quite a clear readout. And we also chose the residues that don't interact with CXCL10 and the receptor at all to make sure we have this accuracy. We obviously determine how each ligand, how each mutant interacts with the ligand first to see that there was no significant changes. And then from then determined the EC80 that was used in the inhibition assays. So when we validated this computationally derived epitope by TruePAP, we could see that a lot of these mutations didn't actually produce quite a significant change. However, there was some differences observed at the N-terminus and ACL2 that shifted the curves slightly to the right. And then when we look at the actual values of the affinity and log alpha beta, it was quite interesting because even if those mutations didn't induce a significant change, all of them trended into the direction of lower functional affinity and lower negative cooperativity compared to the wild type. So when we look at this epitope, we can see that HU37 engages likely large network of residues, and single point mutations are unlikely to cause any significant changes in inhibition. So potentially the idea for future work would be to combine those mutations to see how they really affect the binding of the antibody. So in conclusion, we wanted to point out by the presence of CXCR3B and all the data collected here that existence of GPCR splice variants should not be overlooked in therapeutic approaches. And especially we show that by CXCR3B, where the unique N-terminal extension at this receptor isoforms drove divergent pharmacology, selectivity to the ligand, and it could ultimately be something that determines its physiological function. We also established a very molecular framework of CXCR3B by resolving a 3.4 Armstrong structure of this receptor, which showed that overall the receptor architecture, microswitches, and G-protein interface are quite conserved with CXCR3A, but there were minimal differences observed that could potentially correlate to lower potency that we observed. We also, importantly, showed that CXCR3B is capable of recruiting G-alpha-IO proteins, which contrasts with previous literature, which strongly indicates that this doesn't happen, and this receptor exhibits more atypical chemokine receptor properties, where most of its physiological function happens through beta-arrestin pathways. However, while we show this in our system, it's obviously all artificial. So the next logical step would be to really see what's the functional relevance of this G-alpha-IO signaling pathway at CXCR3B and see how it really contributes to disease progression by looking at endogenous systems. We also showed that MAPs can potentially achieve higher selectivity between receptors through engaging more diverse epitopes, However, obviously, we were unsuccessful with deriving that epitope by Cryo-EM. So importantly, it should be considered that those both isoforms are co-expressed in certain disease contexts, and therapeutic strategies must consider their relative expression, how they interplay with each other, and ultimately what are their context-dependent roles in different disease conditions. So with that, I would like to say a huge thank you, especially to my supervisors, because they obviously provided such a supportive and encouraging atmosphere, especially Nat, who's been there with me from day one and was an incredible and patient teacher throughout the whole experience. As Nat said before, when I came here from the chemistry background, I didn't know how to hold a pipette and I didn't know what the concentration response curve is. So Nat was there with it all for me and patiently taught me all of these, also providing great advice as a scientist and on a personal level throughout this experience. And same goes to Rufio, who's been a great support. She's always been a shoulder to cry on when my cells got contaminated. So if your cells got contaminated, go to Rufio, right there. So yeah, she's been great support and really showed me and taught me ins and outs of pharmacology. Obviously, Greg taught me so much how to evaluate my own work, how to be autonomous and independent as a scientist as well, and provided really valuable advice through the whole experience. And last but not least, of course, Theo joined the supervisory team quite late, but his work in the final stages and help that he put, an effort that he put for the project really ultimately what made my thesis what it is today and really helped, really was patient to teach me all of these challenges of Cryo-EM. So yeah, obviously, huge thank you to my supervisors. I would also like to thank all of the colleagues from MTDD Lab. It was a great environment to work with. I was just really lucky to go to work every day and be excited to just speak to everyone. Collaborators at CXCR3 helped hugely by hosting me for 3 months at their facilities, and there I learned a lot of different things that I couldn't obviously learn at MIPS, just biophysical techniques. Obviously, this wouldn't be possible without the scholarship from Center of Cryo-EM. So obviously, huge thank you to Patrick and Denise and Tracy and Jackie for giving me the opportunity to do my PhD here. Obviously, would like to thank Matt for teaching me Cryo-EM and also being quite patient with all of this. And obviously, all the panel members who advocated for me throughout this experience. And obviously, family and friends. So yeah, thank you so much, and I'll be happy to take your questions. Yes.
I understand that it was there in the prep, it was there in the final exam. So what do you think happened during the dissertation process that prevented, I guess, capturing that concept? And did you try optimizing that process at all, like grades or instrumentation, all that stuff?
Yeah, so the reason I think the complex dissociated could be because of the high fast off rates of the antibody itself. We actually tried to do that at CXCR3 by measuring the dissociation and association rates of this antibody. So that's one possibility. Another possibility is just simply the pressure of the air-water interface, where obviously the air-water interface would destabilize the complex lead to dissociation. We did quite a lot of things to optimize it, mainly from the biochemistry standpoint. We did try purifying it in the presence of nanobody 6, small molecules. We tried using fab fragments and all of that, and it didn't really help. So at some point decided to pivot and try to establish this epitope in more of a different technique. So yeah. Yes.
In your CXCR3 pharmacology, the concentration response curves look very biphasic to me, at least from this perspective. Did you look at analyzing those with a biphasic model, and what do you think is going on?
We didn't analyze it with the biphasic model, but it is quite possible that there is different populations of the receptor possible that it can homodimerize, heterodimerize. There's some spectruciferase assays that show evidence for this. So it's just possible that different populations that signal differently. But yeah, we didn't do any phase models. Is someone on Zoom trying to ask a question? Okay, no questions on Zoom. Is there? Yeah.
Nice talk and congratulations.
Thank you.
And just a more general question, because like CXCR3 is related to cancer, so and your approach profiling is focused on the GIO family. So does this receptor also coupled to other key proteins like GXCR13 or some other subtypes?
So there was early reports from like 2003 that suggested that maybe it couples to G-alpha-S, but it was disputed in our lab and also in a literature that came out later on. So that's why we focus on G-alpha-IO, and literature assumed that the anti-proliferative function actually comes from arrestin signaling, or is likely to come from that. So yeah, we don't really know how G-alpha-IO contributes to it. So it's all cell type dependent as well, but yeah, we didn't. We also tried G-alpha-Q actually, because there's reports of both receptors stimulating some calcium, but we didn't see any response. So that calcium response could also come from G-alpha-I itself. Yes.
My question is around the ligands. So using CXCR11 in your structure, and then you didn't see it in the final structure. Were you using exogenous ligand? Did you express it with the receptor, or yeah, what was the process around this?
We purchased it. So basically, we tried to express it in BL21 cells. I lost it all in the solution, and I needed inclusion bodies, and that required optimization that was too time consuming. So we just purchased it. It wasn't cheap, but yeah, it helped. Like, what do you mean by co-expressed is? Not really. We also tried to avoid doing that to just make sure that the interactions that we observe, if we did see the ligand, were native. So we tried to not, do any fake cross-linking or things like that. But yeah, I think there is a question on Zoom.
Yeah, it's me, Isabel. Yeah, absolutely great talk, and thank you, very clear. Is there any glycosylation that could be an issue, and that could explain differential binding?
So no glycosylation. This receptor was thought to, in terms of post-translational mutation, have sulfation at the N-terminus. That was thought to influence the binding of the ligand. But the recent Cryo-EM structures and also mass spec showed that this didn't happen. We actually tried to do mass spec on this receptor, but we didn't really see much beyond the N-terminus. So it was great, at least somewhere we saw the N-terminus, but we didn't see any significant PTMs.
Yeah, thank you.
Yes. Cool. All right, thank you all.
Okay.